Hello everybody. Today's topic of discussion is regulation of gene expression. Well, first of all, what is gene? Gene is a portion of DNA responsible for synthesis of one polypeptide chain. Then synthesis of protein from a gene is called gene expression. On the basis of gene expression, structural genes are classified into two. One is constitutive genes, other is inducible gene. Constitutive genes, another name of constitutive genes is housekeeping genes. Their genes are expressed at a more or less constant rate in all cells in our body. That is, they are not subjected to regulation. Example of constitutive genes are enzymes of glycolysis, TCA cycle. Then the inducible genes. They are induced or repressed as per need of the metabolism. Such regulation is done by induction and repression mechanism. Example, repression by hemoglobin on allosynthes. Then there are two types of gene expression. One is positive gene expression and second is negative gene expression. What is positive gene expression? That is induction. When gene expression is increased by a inducer, is called induction. And when the gene expression is decreased by a repressor, it is called negative gene expression or repression mechanism. The gene expression mechanism can be explained by operon concept. The concept of operon was introduced by Francois Jacob and Jacques Monod in 1961, and they were awarded Nobel Prize in 1965 based on their observation on the regulation of lactose metabolism in E. coli. Then, what is operon? Operon is the coordinated unit of genetic expression in bacteria. Operon consists of cluster of structural genes regulated by a common promoter regulation. Then, structure of a lac operon. It has got four parts. One is regulatory gene, that is I. Second is promoter site, that is P. Third is operator gene, that is O. And the structural genes Z, Y and A. First of all, the regulatory genes. The expression of this gene is constitutive. From this gene, repressor molecules are synthesized. Then the promoter site, it is the binding site for RNA polymerase. Then the operator gene, it is the binding site for repressor molecule. Next, the structural gene, that is the first is Z gene, that is responsible for synthesis of beta galactosidase enzyme. The function of beta galactosidase is it hydrolyzes lactose into galactose and glucose. Then the Y gene, that synthesizes the galactoside permease enzyme. The function of this enzyme is it transports the lactose into the cell. Then the A gene, that is the responsible for synthesis of thiogalactoside transacetylase enzyme. But the function of this enzyme is not known. And the picture shows from 5' to 3' the order of genes are present. That is first the I gene, that is the regulatory gene. Second is P gene, that is the promoter site. that is then the O gene, that is the operator site, then the 3 structural gene, that is Z, Y and A. Then the repressor molecule. Repressor molecule has the 4 identical subunit and they have a strong affinity to the operator site. Then how the gene expression, regulation of gene expression takes place. When the lactose is absent in the medium, what happened? Regulator genes produce the repressor molecule. Because the expression is constitutive, these repressor molecules binds to the operator site. So the RNA polymerase that binds to the promoter region will not move to the structural gene. This is called repression of the structural gene. Thus, when lactose is not available, the lactose utilizing enzymes are not synthesized. The picture shows from I gene, that is regulatory gene, mRNA is synthesized, and from this mRNA the repressor molecule are synthesized. Then the repressor molecule has got a high affinity to bind the operator site, and the repressor molecule binds to the operator site. RNA polymerase that is remain binds with the promoter site cannot move to the structural gene. So no lactose metabolizing enzymes are synthesized. Then when the lactose is present in the medium, what happens? When lactose is present in the medium, lactose enters into the cells with the help of galactoside permease. Then the beta galactosidase enzyme isomerizes some lactose molecule into the allolactose, and rest the molecule of lactose are hydrolyzed to form galactose and glucose. Then the allolactose isomer of lactose gets binds with the repressor molecule that is produced by the regulatory gene and gets inactivated, which do not binds to the operator region. So there is no repressor molecule at the operator site. Now the RNA polymerase can transcribe the structural gene, which are then translated. Lactose induces the synthesis of lactose utilizing enzyme. Hence the lactose is an inducer of this gene, and the mechanism is said to be de-repression of the gene. The picture shows when the lactose present in the medium, lactose enters into the cells by the permease enzyme. Then by the enzyme beta galactosidase, the lactose within the cells, some molecule of lactose isomerized to form allolactose, and rest of the molecules are hydrolyzed to form galactose and glucose. Then the allolactose molecule binds with a repressor molecule, that is the repressor molecule. The repressor molecule are synthesized from the regulatory gene. Then this allolactose and repressor molecule, combination of repressor and allolactose molecule cannot bind to the operator region. So the RNA polymerase can move to the structural gene, produce the mRNA, and from this mRNA the protein Z, Y and A proteins are synthesized, that is the lactose metabolizing enzymes are synthesized. So the lactose are metabolized. Then when both the glucose and lactose are present in the medium, when both the glucose and lactose are present in the bacteria, the bacteria preferentially utilize the glucose. When glucose is exhausted, then the lactose is metabolized. In presence of glucose, as the glucose inhibits the adenyl cyclase, the concentration of cyclic AMP will decrease. There is a decreased cyclic AMP cap complex formation. CAP means, full form of CAP is catabolite gene activator protein. So there is a decreased binding of RNA polymerase to the promoter region, because the cyclic AMP CAP complex is required to bind the RNA polymerase to the promoter region. Then the decreased synthesis of lactose metabolized enzyme. Then when the glucose is exhausted, there is increased cyclic AMP concentration, increased formation of cyclic AMP CAP complex, increased binding of RNA polymerase to the promoter region, increased synthesis of lactose metabolized enzyme. Then the gratuitous induction. What are they? Certain structural analog of allolactose that de-repress the structural gene, that leads to increased synthesis of lactose metabolizing enzyme, but not substrates for this enzyme. Example is isopropyl thiogalactoside, that is in short IPTG. Then the clinical application. Repression is the mechanism by which the presence of excess product of a pathway shut off the synthesis of key enzyme of that pathway. Heme synthesis is an example. It is regulated by repression of the ALA synthase. The key enzyme of the pathway, regulatory gene, produce the aporepressor, which binds with the heme and becomes the active holorepressor. Here the heme acts as a co-repressor. The holorepressor binds to the operator, so the RNA polymerase cannot move to the structural gene and the enzyme synthesis will stop. The picture shows regulatory genes constitutively express the aporepressor molecule. Then aporepressor molecule binds to the co-repressor molecule, that is the heme, when heme is present in the medium, and this holorepressor molecule binds to the operator region, so the RNA polymerase that is binds with the promoter site cannot move to the structural gene, so the ALA synthesis will decrease. That's all for today. Thank you.